I have a task to cover the HPV biology, or revisit HPV biology, and in the perspective for cure. So you've heard quite a lot of different talks up to now. So I don't know if there will be new information, but I will try to give you at least my point of view on that point. In terms of what we want to achieve in terms of cure, you heard different talks even this morning. What I think there is a consensus now is that what we want to achieve on the short term is a functional cure that is defined by the loss of HBs antigen after treatment cessation that would be sustained after this treatment cessation, that would be still associated with the persistence of cccDNA in the liver and integrated viral DNA form. There's a consensus that this would be the most attainable goal in the near future. So then we can think of more ambitious goals, such as complete cure, where cccDNA would be eradicated, but still with the persistence of integrated forms. And then there would be the real optimal goal, which is the sterilizing cure, where we would lose all the viral DNA forms, including cccDNA and integrated forms. Now, regarding the HPV life cycle, you all know the main steps of this replication cycle, and you know that the current drugs, interferon alpha, is mainly working on the stimulation of the innate immunity. It has some effect on cccDNA, either by silencing or doing some degradation of the existing pool, but in vivo, in patients, the overall antiviral effect is really marginal. Nucleoside analogs, they block viral replication, but they are only suppressive. They don't have a direct effect on cccDNA, so we need lifelong therapy. In some of the patients, the viral suppression may be associated to some restoration of adaptive immunity. But this is really also a marginal effect on the minority of patients. So we really need to do much more than what we have if we want to achieve a functional cure in many more patients. Now, what are the barriers to eradicating HPV? We can split schematically the issue in two parts. One is the viral determinants, and the main one is the persistence of cccDNA. And you know that there's a long half-life. There's a continuous replenishment by either the recycling of nucleocapsid within the infected cells, or by infection of new cells. And this cccDNA reservoir is not directly affected by nucleoside analogs and really minimally affected by interferon. And we have also the persistence of integrated forms, and we had a debate yesterday about it, which lead to viral persistence. Now, there's this immune counterpart, and I won't go through in detail because this was addressed yesterday by beautiful talks. And we know that in chronically infected patients, these patients have defective B cells and CD8 positive responses, and there's also inefficient innate responses in these patients. So overall, there are defective immune responses, and all this together lead to HPV persistence. And we have really hard time either to eradicate the cccDNA reservoir, or to restore specific immunity. Now, I would like to really focus my talk on cccDNA and try to see what we could do to control or even eradicate cccDNA without killing the hepatocyte. So here we will not address the issue of immunology. So a little bit of remind us here that cccDNA persists even in patients who are going to clear HBs antigen even after HBs seroconversion, which is defined as a functional cure. Here's just an example of a patient who received sequential nucleoside analog therapy over time, and finally he was able to clear HBs antigen, had even a seroconversion to anti-S antibody to very high titer, 1,000 international unit per ml. And we had the opportunity in these patients to have sequential liver biopsy performed. And you see here the decline of intrahepatic viral DNA in the liver of these patients, and cccDNA is in black, and you see that even at the time of anti-S seroconversion, so we had a decline by approximately 3 log of cccDNA in the liver, but there was still persistence of cccDNA in this patient. So this is just one example. There are many others that show that actually, even after resolution of infection, cccDNA persists. And so HBV is a real model for persistence. Here, a clinical case that was just reported in hepatology recently. It's a work we've done with Darius Moratpour, where he had a patient who had a late hepatitis B reactivation following anti-HCV treatment for a recurrent hepatitis C on the liver transplantation. And this patient was anti-HBC positive. That was the only marker of HBV infection that he had prior to transplantation. He had a full active HCV replication, was transplanted for HCV disease. Then HCV was recurrent on the liver graft with a progressive liver disease, and the patient received sofosbuvir, ledipasvir therapy. So you see here HCV RNA declining, then HCV was cleared from this patient. But what was shown is that 30 weeks after the end of the HCV treatment, HBs antigen became positive again with very high viral load. And then the patient was treated with tenofovir and was then in viral suppression. But then what we did is we went back to the liver explant, did some sequencing of HBV DNA in the liver explant, and compared to the circulating virus in the blood circulation at the time of the relapse. And they were actually matching, the sequences were matching, suggesting that it was really a reactivation of the virus, of HBV, following HCV clearance. Another important clinical aspect is the fact that even during long-term tenofovir therapy, and here it's a cohort of co-infected patients, HIV, HBV, co-infected patients who receive long-term tenofovir therapy. And in that cohort, obviously all patients had undetectable HBV DNA because of the of tenofovir treatment. But when you look at the serum compartment here, HBs antigen quantification showed that HBs was plateauing, which is well known. But we had the opportunity to have paired biopsy samples in some of these patients after several years of treatment. And what we could show is that in these patients, despite I would say an effective tenofovir therapy with undetectable HBV DNA in serum, there was still HBV DNA synthesis in the liver of this patient that may explain the persistence of cccDNA, either by new round of infection or by replenishment of the cccDNA pool within the infected hepatocytes. So now if we want to target cccDNA, which is really the cornerstone of our persistence regarding the viral mechanism, there are several ways, and you heard about the different paths that we could follow to target cccDNA. We could target cccDNA formation, try to induce degradation, or to silence cccDNA if we cannot eradicate cccDNA completely, or induce a loss of cccDNA through hepatocyte turnover. So these are the, I would say, ways to directly target cccDNA, but there are other ways, indirect ways, which are to try to decrease cccDNA replenishment, either by preventing new round of infection or by preventing the cccDNA replenishment from the recycling of nucleocapsid within the infected cells. So regarding these indirect ways of targeting cccDNA, there are the entry inhibitors to prevent new rounds of infection, and you all know the great work performed by Stefan Urban's group since many years now, which actually was started before the identification of the receptor NTCP, but then was accelerated after that discovery, and the Mircludex, the PS1 peptide mimicking the PS1 domain interacting with NTCP is really one of the way to block viral entry. But the problem here is that to induce a decline in the pool of cccDNA would need to have quite a significant cell turnover to have an effect on that pool of cccDNA, which is not really the case in patients that are controlled with NUCs. Now there are other ways to target cccDNA indirectly by the replenishment is with the core inhibitors, and there's a lot of capsid assembly modulators that are being developed by several companies. And here I'm just showing you one example that was published recently, where they showed here that core inhibitors not only inhibit viral genome replication, which is the first effect you would expect by interfering with nucleocapsid assembly, but it does also prevent cccDNA formation when administered prior to HPV inoculation here in this experiment. So that would be also a way to prevent or decrease the new rounds of infection in the infected liver. Whether the capsid assembly modulators interfere with the recycling of the cccDNA within infected hepatocyte has not been clearly demonstrated, but we could infer from this data that this is the case. So here are the would say the two mode of action of these capsid inhibitors. Here, inhibiting viral genome replication by interfering with nucleocapsid assembly, and here by preventing cccDNA formation from the incoming nucleocapsid in the nucleus of infected cells. Now, let's see the direct targeting of cccDNA. What we like to do is obviously induce a degradation of cccDNA. The first proof of concept was given by the work of Ulla Protter's group, showing that interferon alpha and levotoxin beta can induce an ApoB-dependent degradation of cccDNA. The following work also showed that similar observations were done with interferon gamma and TNF alpha. The caveat here is that This was demonstrated in experimental model in tissue culture, so we are far from the clinical situation, and there was not a complete clearance of the pool of cccDNA. But that was, I think, the first proof of concept that paved the way for new works in the future. Now, you've just heard a few minutes ago that we can target HBV with gene editing approach, such as the CRISPR-Cas9. And here I'm just summarizing a work, it was a work done by Christoph Zieger's group in Philadelphia, where he showed that using CRISPR-Cas9 in cell culture experiment, it could induce mutations and deletions that lead to functional inactivation of cccDNA. And here you see the immunostaining for core antigen in these cells that were treated with the CRISPR-Cas9 approach. So, and then they made an extensive study to show that over 90% of HPV DNA was cleaved by Cas9, and it compared to interferon treatment, and showed that Cas9 cleavage was 15,000 times more efficient than the ApoB-mediated deamination following interferon treatment in this cell culture model. So this is something that is very interesting. We know all the hurdles that we have to face with CRISPR, but it's a beginning of the story, and I think this will be very important to follow in the future. So the other approach to induce a loss of cccDNA would be to induce hepatocyte turnover, and this was shown very nicely by the group of Maura Dandry some years ago when they did partial hepatectomy of this humanized mice and induced cell turnover, and showed that there was a decline of cccDNA in these mice. So this is something that is nice experimentally, but in patients, I wouldn't recommend to go in that direction, because that would trigger some carcinogenic event, especially if the hepatocytes that are turning over contain integrated sequences. Now, there have been also a lot of questions around whether we could follow cccDNA, the fate of cccDNA during the infection. So several works, and here I'm just showing a very recent one from Christoph Zieger that's just been published, where he tried to establish a fish technology to localize cccDNA and follow the fate of cccDNA during infection and nucleoside analog therapy. So you see here all this setting up of the experimental condition for the fish analysis, and then they did nucleoside analog therapy and cell passages, and they showed that the reduction of nuclear DNA was consistent with a symmetrical distribution to daughter cells, because this was still a debate whether it would be symmetrical or non-symmetrical when you have hepatocyte mitosis. Now, if we go to cccDNA formation and potential targets, you see here the main steps of cccDNA formation from the incoming capsid in the nucleus of infected cells, and where the capsid will release rcDNA, and then there will be several steps, biochemical steps to form cccDNA that include the removal of the polymerase from minus strand DNA, the removal of the viral RNA from plus strand DNA, the completion of plus strand DNA, and ligation to form a protein-free rcDNA, and then a complete cccDNA. So these steps involve many enzymatic pathways that depend on the host, and this was nicely shown by Michael Nassal's group a couple of years ago, where he showed that involvement of TDP2 to cleave the phosphodiester bound from the polymerase to -strand DNA. But later on, it was shown that in cells that were knocked out for TDP2, you could still have a cccDNA formation, implying that many other nuclear enzymes could be involved in this reaction. So we still a lot of work to do to better understand what are the DNA repair mechanism involved in cccDNA formation that could be very important in the future for future therapies. So in that respect, we were quite interested in lab to understand how does it work? I mean, in terms of the initial step of cccDNA formation. We need obviously DNA repair. And cccDNA is a mini chromosome, so we also need chromatin to be deposited on cccDNA. So the question was, are DNA repair and chromatinization coupled in the formation of cccDNA? So we had, if you go back to cell biology and regarding chromatin structure, you know that the nucleosomes are formed by different histones, mainly histones H3, 2A, 2B, and H4. And you have also the H1 histone that serve as a linker DNA between the different nucleosomes. And there's a specific dynamics, and Steve Lucarnini told you about this morning, of the chromatin structure, and depending on genomic regions, whether it's centromeres, telomeres, or genes, and depending also on DNA processes, whether the cell is in replicative states or whether there's DNA repair or transcription. And we know that, for instance, one of the histone variant, H3.3, is a DNA replication independent histone variant, and it replaces conventional H3 in a wide range of nucleosomes in active genes. And for that, all these specific dynamics, there's a role of histone chaperones. And one of these chaperones, among others, is IRA. And this chaperone is involved in DNA repair. And it is known to recognize naked DNA to deposit histone H3.3. And IRA is responsible for chromatin priming in response to DNA damage, allowing transcription recovery after repair. So there's quite some similarity with, when you think of it, in the formation of rcDNA to cccDNA, that would involve a DNA repair mechanism and then a transcription from the cccDNA. So we ask whether IRA is involved in cccDNA establishment to allow the histone H3.3 deposition. So to do this, we set up a cell culture model including HBG2 NTCP cells and primary hepatocytes, and look at the early kinetics, because we wanted to look at the early events of cccDNA formation. And we showed that by qPCR analysis that the cccDNA could be detected almost 2 hours post-inoculation. It could be confirmed with a little bit delayed with a southern blot analysis because of the different sensitivity of the assay. After 1 day post-inoculation, cccDNA could be detected. So what we did here was to use a chromatin immunoprecipitation on infected cells and see whether we could find IRA and the different histones on cccDNA. So to make a long story short, what we showed here is that IRA could be found on on cccDNA early after infection of these hepatocytes, and this was associated with the deposition of the H3.3 histone variant, and that was correlated with the initiation of viral transcription from cccDNA. So then what we did next is to do silencing of IRA. So we use a siRNA to silence IRA, and we found that silencing of IRA decreased cccDNA levels either by thousand blotting or by qPCR assays. At this stage, we could rule out the role of HBX in this process, as when we used X-deficient virus, there was no difference in observation we had. Currently, we are investigating whether the capsid protein could play a role in the recruitment of IRA on cccDNA. So this work is still ongoing and may be important in the future. So if we come back now to I would say, a steady state infection model where hepatocytes are chronically infected, as you know, and Stephen showed that this morning, I mean, cccDNA is really present in the cell as a viral mini chromosome, and depending on the post-translational modification of histones, cccDNA can be either in a very compact form or in a more open form that is then susceptible to transcription activity. And here, if we think of therapy, here we are dealing with strategies that would silence cccDNA, so either using epigenome modifiers, interferon alpha, capsid inhibitors, HBX inhibitors, as it was said this morning. But we need to better understand what are the viral effectors of the cccDNA chromatin status to be able to develop strategies that would be viral specific and not to avoid off-target effects if we are using epigenome modifiers, for instance. And here you see a summary slide that showed that interferon alpha in different systems was shown to induce a repression of viral transcription from cccDNA by inducing post-translational modification of histones. So we are also interested in the lab to know better, to have a better knowledge of what is the status of the chromatin associated with cccDNA, as this may have very important implications, and we heard this morning about CRISPR-Cas9 accessibility, for instance, or by identifying specific targets for future therapies. So here what we did in this setting was chronically infected hepatocytes, either HBG2 NTCP cells or primary human hepatocytes. We did the chromatin proteomics to identify the partners of the viral capsid protein that is bound to cccDNA. And what we found that we found more than 300 proteins that were identified in either HBG2 or PHH cells. But what was interesting is that the pattern of the proteome was different between the HBG2 NTCPs and primary human hepatocytes. So we are now focusing on these partners that were identified in primary human hepatocytes to better understand their biology and their role in cccDNA biology. So we did some gene ontology analysis of this cccDNA-associated proteins, and you see here the main families of proteins that were found, either involved in chromatin structure, chromatin modification, or transcription, which was in the end a little bit, would say, expected. And then we went for the selected partners to do some inactivation by siRNA to see what is the phenotype of this knockdown, and we could show that for some of the partners that are still under analysis, that we could see a decline of cccDNA and a decline of viral RNA, including pregenomic RNA, in the cells that were treated with siRNA. So this work is still ongoing, but may lead to a very interesting finding regarding the accessibility of cccDNA to other approaches for gene editing or for identifying specific interactants with the capsid protein that may be very important to develop novel strategies. So the challenges in targeting cccDNA are really to find specific approaches that wouldn't interfere with the cellular biology. So I won't go through in detail, but that's really the main point here. So we need to really put more effort in terms of research in the cccDNA biology if we want to really have make a dramatic change in our knowledge here. So if we then think of the clinic, go back to the clinic now, is the fact that whatever drugs we'll be developing, direct-acting antivirals, immunotherapeutic drugs, we need to have to assess the reservoir of cccDNA in the liver. And we are doing, as clinicians, less and less liver biopsies. So we will need to have novel biomarkers to assist drug development, and we discussed that during the past 2 days. So there are efforts first to really try to standardize cccDNA assays, and you heard about it. So there's an international concerted action with ICE, NRS, and DZIF on that, really to make sure that we are using the best assays we can, and that we speak about the same terminology and the technology to assess the effect of different strategies on cccDNA. But if we go then to the clinic, we will need to have really biomarkers in the blood circulation. So what is discussed now is circulating viral RNA and HBC-related antigen, because we know that quantitative HBs antigen is not a very good, reliable marker, because it can come either from cccDNA or integrated forms. So to conclude now, the last part of my talk is really as a clinician, whether we can cure both the infection and the diseased liver, and this was addressed yesterday in the debate. And clearly here is whether we are coming too late, even if we cure the infection, if we cure cccDNA, are we going to be too late because there's already the integration that will lead to molecular events that will lead to liver cancer later on? So well, I will go fast there because you've seen these slides many times. So the whole rationale around that was that we've seen slower HBS decline in HBe negative patients for many years, whether they were receiving nucleoside analogs here compared to E positive patients or interferon. And then we got the newest results with siRNA where it was shown by Howard that the decline of HBs antigen was much slower in the E-negative population, maybe because, most likely because of the integration. And here it's just a summary slide that you've seen before, clearly stating that HBs antigen is really expressed from both cccDNA and viral integrant, and this may be even more true when patients have long-lasting disease and in the E-negative phase. And the integration in the host chromosome is seen at very early stage in young patients, as was shown by the Bill Mason's and Antonio Bertolitti's work, even in the so-called immune-tolerant patients at very young age. So you can see integration and the clonal expansion of hepatocytes in all disease phases, so, which is really an argument that we will need to, whatever treatment we'll have, to go for very early treatment intervention, not wait for all these integration events to occur with inflammation, fibrosis, cell turnover. And integration, we will have to take care of it at some point. Maybe it's too early now, but because we don't have the tools or the technology, unless maybe siRNA may be an indirect way to contribute to an HBs decline and try to restore some T-cell activity because of this HBs expression. This has to be seen, but we will need to address that in the future. So in conclusion, I think we are not trying to, there's a lot of effort to identify really novel targets for direct antivirals, and I'm pretty sure that in the future, either directly or in combination with immunomodulatory strategies, we will achieve better results than what we have now. The main question will be whether we will be able to implement strategies at the global population to really treat early, intervene early, to prevent cirrhosis and prevent liver cancer. And with that, I will acknowledge my collaborators in Lyon, the pathology unit, INSERM unit, and also make an advertisement. I'm not the only ones that did it. So we'll have the HPV Cure Workshop organized by NRS. That will be the 5th one in Paris, just before the EASL meeting, the day before. So you will be welcome to attend. Thank you.